going to be talking to you about the genetic variability of flowering and nectar traits in Tasmanian Leptospermum, specifically in relation to the production of medical grade honey. So what makes Leptospermum honey so unique? Well, Leptospermum nectar has a unique compound in it called dihydroxyacetone, or DHA for short. And that DHA is converted to methyl glyoxal in the honey, or MgO. And the higher the MgO, the higher the antibacterial activity and the higher the value. You would have all seen MgO ratings on labels in the supermarkets. So what leptospermum resources do we have here in Australia? Australia has at least 83 leptospermum species, including the species called Leptospermum scoparium that is used to make the well-known Manuka honey. And we have 8 of those leptospermum species growing in Tasmania, and 6 of them are endemic, meaning they only grow there. So there's a unique resource in Tasmania that can be utilised. And previous research has indicated that the DHA levels in nectar from some of these Tasmanian species are equivalent to or higher than that found in Leptospermum scoparium. And this includes Lenigerum, Riparium, Nitidum and Rupestre. Now it's these Leptospermum species that I am studying in my research project. Now, if we want to produce consistent Leptospermum honey, it's proposed to use stationary plantations in order to achieve a number of breeding objectives. Now, what are some of those breeding objectives? Firstly, we want to maximise nectar DHA levels, to maximise the MgO levels in the honey. Secondly, and very importantly, we want to achieve consistent levels of DHA every year to achieve a consistent honey product. And thirdly, we'd like to maximise the plant nectar production so that we can get a high honey volume produced. Now, if we want to achieve those breeding objectives, one of the things we need to understand is what's the genetic variability of the relevant plant traits to those breeding objectives. Does it matter where we select plants from in Tasmania for breeding, or is there no difference at all in the expression of those traits in the different areas of Tasmania? And how much does the plant trait expression affected by genetic versus environmental factors? So the aim of my research project is to study the genetic variability of these key traits which impact on the production of medical grade honey within Tasmanian Leptospermum species. Now, what are the traits of interest? Firstly, nectar DHA levels, quite importantly. Secondly, total nectar sugar concentration, because that can be a bit of a surrogate measure for the likely volume of nectar we're getting from a plant. And then we want to look at some flower production and flower duration traits, because that will influence the amount of nectar produced by the plants. And then when establishing a plantation, we're also interested in plant survival and growth. So the key objective of my research project is to characterise the genetic differences between populations and families of Leptospermum in Tasmania for these traits of interest. Now, what do I mean by populations and families? So if you have a look at the map of Tasmania here, the small green dots are the relative distribution of Leptospermum scoparium throughout the state. Now, prior to my research project starting, Anthony O'Grady from the CSIRO spent about 4 and 5 years travelling around the state collecting samples of Leptospermum from the different locations represented by the yellow and orange dots. And at each of those locations he collected nectar from the mother plants, seed and cuttings. Now each of those locations would be what we would consider a population. So there's a genetic variability we can look at there between populations throughout the state. Now at each of those population locations, Anthony O'Grady also collected material from at least 5 plants. Now it's the seedlings produced from the seed from those 5 plants that would be considered families, or a within population variation. So there's 2 levels of genetic variability that we can look at here for breeding objectives, and that is between populations and within populations, or at the family level. But how do we study that level of genetic variation when there's also different environmental conditions at each of these population locations? Well, the way to do that is to grow seedlings from the seed collected at each of these locations and plant them all in one location where the environmental conditions are the same. And prior to my research project starting, Anthony O'Grady did this at the Hobart field site using the seed collected from all the locations represented by the yellow dots. So by studying the expression of the traits at that site, we can see that if there is a significant difference between the populations or the families, we know that that expression of those traits is under high genetic control. But to also answer the environmental question, whether the environmental conditions have an impact on the expression of these traits, I set up a second plantation site during my research project. So I took the same seed that Anthony O'Grady used to set up the Hobart field site, and I grew a whole lot more seedlings and I planted them at Douglas River. Now, by looking at the expression of the different traits at the two sites, if they are similar, then we can see that the genetic control over those traits is high. But if they differ, then we can tell that there is some environmental component to the expression of these traits. So there'll be some genetic bioenvironmental interaction. So today, the data that I'm going to be showing you is just looking at the initial genetic variability question from the data collected at the Hobart field site. The second aspect of the environmental side of things, that data has been collected but is currently being analysed. So here's a bit more detailed information about those two plantation sites. You can see on the left-hand side of the slide there a couple of pictures. The top picture is the Hobart plantation site as it currently stands now with the mature plants, and the bottom picture is an aerial view of the Douglas River plantation. Now, the bulk of the plants at both of these plantations is Leptospermum scoparium. However, the Douglas River site does include some of the other Leptospermum species from Tasmania in the form of clones to answer some other aspects in the research. Now, because the Hobart field site was established earlier, the plants were more mature, which meant I could collect nectar samples earlier and other data. So the bulk of the data for my research project is coming from here, and that is the results that I will be presenting to you in the next slide on the genetic variability question. So just to give you an idea of the type of data being collected at these two sites, data is being collected at the Hobart plantation for the last 3.5 years and for the Douglas River plantation the last 2 years, and that data will continue to be collected after my research project has finished because there's plenty more research that can continue in future years. Now, what types of data have been collected? So a flowering assessment has been conducted monthly, except in the specific summer flowering season that's shortened to fortnightly because the plants themselves have about a 2-week flowering period. Plant growth or height is collected 6-monthly, survival and health monthly. And then importantly, with nectar samples, they're collected daily during a flowering season. So each day we'd go out into the plantation and look for new plants that are flowering. And to date, in the last 2 flowering seasons, over 2,000 nectar samples have been collected. So with such a high number of nectar samples that need to be analysed, one of the first things I needed to do in my research project was to develop a new nectar analysis method. So in collaboration with David Nichols at UTAS, I developed a new analysis method for quantifying DHA and sugars in the Leptospermum nectar using ultra-high pressure liquid chromatography and mass spectrometry. And there are a number of improvements that were achieved with this new method. Firstly, there was a significant reduction in cost per sample, which allowed the thousands of samples to be analysed within the project budget. There's also a significant reduction in sample preparation time, which is very handy when analysing so many samples. There was improved limits of detection and repeatability, so it was a very robust method, and I was able to detect very low levels of DHA and sugars. And then lastly, this method allowed the identification of individual sugar types of fructose, glucose and sucrose, whereas the previous methods only allowed the total sugars to be analysed. So this table here provides a summary of all the data collected at the Hobart plantation site on the Leptospermum scoparium to answer that genetic control question. So if you look at the left-hand column of this table, I'll just go through the different plant traits that were analysed. And this is just a small summary of some of the traits because of timing constraints today. So the first trait, the DHA to turtle sugars ratio, that's a common ratio used in the industry to help predict the type of MGO levels you're going to get in the honey. Then there's nectar total sugars, which I've talked about before. Growth, which is the height of the plants. The next trait is age to first flowering, and that's based on once the plant is established in the plantation, how many days it takes before that plant first flowers. So that gives you a good idea of how quickly a plantation may start producing nectar. The next trait, time to peak flowering, that's looking at within a specific flowering season, how long a plant takes in the number of days before it gets to peak flowering period. The next trait, maximum flowering intensity, that's looking at the number of flowers on a plant in proportion to the size of that plant. And duration of flowering is how long a plant flowers for within a season. So those last two traits are really looking at increasing nectar production in a plantation. Now, if you have a look at the next two columns in this table under the title significance, this is giving you the results for whether there's a significant difference between populations and between families when looking at the expression of these traits on a genetic level. And if there's a star, that indicates a significant difference. And the number of stars provided indicates the size of the significant difference. And if there's an NS, that's no significant difference. So really importantly, you can see that in that first column under the between populations difference, every single trait has a highly significant difference at the population level. So there's some genetic variability we can capitalise on there in our breeding program, which is really good news. And if you look at the next column, the family within population differences, there's still some traits there that have a highly significant difference, especially if you look at that first trait, the DHA to total sugars ratio, which is really important for getting that high DHA level in a plantation. We've got genetic variability there at the between population level and within population level that we can capitalise on. If looking at the last column under heritability, this is looking at the within population heritability. So this is the proportion of variability within a population that is controlled at a genetic level. The heritability value goes from 0 to 1. So if you're looking at that first value in red, a value of 0.52 means that 52% of the variability we're seeing there is controlled on a genetic level. And that 0.52 is considered a really high, quite high value. And the same again for the 0.62 value for the time to peak flowering further down in red. And then the value in orange of 0.31 for age to first flowering is also considered high. And then for growth, the value in green there of 0.27 is considered a moderate level of heritability. So in summary, from the data I presented to you today, we can see that all Leptospermum scoparium traits studied exhibit high genetic variability. That means the production of medical grade honey can potentially be improved through plant selection and breeding programs. So we have a resource there in breeding. So what are some of the next things to consider? Well, I've already mentioned to you that the other part of my research project is considering the genotype bioenvironmental interaction, and that data has been analysed now. So hopefully in the future, I'll have that information to present. And then beyond my research project, it's really important to look at the plantation economics and silviculture to see if a plantation will work economically and produce the honey that we're trying to produce. So I envisage that would involve establishing a pilot trial site using some selected plants with the traits of interest and proving that we can produce a high MGO honey and proving that it's economically viable. So thank you for your time. I'll just leave up here now while we're having questions, a slide acknowledging all the people that have helped me during this project.